Okay, so the recording's on. So what I was saying before I turned the recording on is this is such a fascinating thing for me to talk about. I love talking about this history of life on Earth. I mean, you look at this thing that looks perfectly round, and to me as a kid, I always sort of remember thinking, man, it's just floating. How does this big heavy thing just kind of float in all this black? You know, this isn't an astronomy course. This isn't, you know, a physics course or anything like that. But I've always been sort of kind of side interested in astronomy too, the development of the planet. So I like to throw that into the mix when I talk about biology. So at the very beginning, if you think about it, 4.6 billion years ago, the solar system was just coming into formation. And what was basically happening is these pieces of matter, like rocks basically, were starting to stick together. They were getting bigger, and they were making planets. And some of them get very big, they stayed like asteroids, and some got really big and started this reaction, you know, that became a star. So, you know, us, the planet here, we were involved in all that too. We were just in this part of the, oh boy, this part of the universe, this part of the galaxy, you know, this part of the solar system. And so as the Earth, you know, developed, you know, from this really hot molten rock, because the early solar system with all those rocks kind of whipping around, going around in a circle, there was lots of bump. It was like a pinball gallery. There's a lot of stuff bumping off each other, like bam, bam, bam. And asteroid collisions, like huge asteroid collisions, like, you know, planet-sized asteroids, running into each other were very common, and that was releasing a lot of energy. Hold on a second. That was releasing a lot of energy, and these rocks were coalescing. And I mean, but it was a rough place. It was not a place for living things, especially in our part of the universe. It was definitely not a place for living things. But then the Earth changed. I mean, it had to have changed, or we wouldn't be here talking about how it changed. So that's kind of the overview of what we're going to do in this chapter. Is we're going to talk about from the beginning when the that sounds very anyway. But we're going to talk about how at the start, how did we go from this rock, from this cooling rock, to this really lush planet like you see in the picture here? You know, the clouds and you see this is the African continent. And it's very clear where the deserts are. You know, the tropical rainforests are, and not to mention, you know, the oceans. We all know how rich the oceans are too in terms of organisms and diversity. And I mean, again, it's just mind-boggling to me. So, you know, we're going to discuss all that as the semester goes along. But what we're going to do today really is kind of put them in perspective as to who came along first, and kind of how that all fit with what was going on Earth. But you know, before we actually get into the living things, we're gonna break up this chapter into two parts. The first part we're going to talk about is this biogenesis, and biogenesis is just what it says, right? It's how did life begin? How did non-living become living? It's hard enough to define what that is, living and non-living, for society in general. But then to actually try to figure when it began, or, you know, when it's not, you know. So this is something that, you know, I'm going to go through these four major ideas that are out there right now, or four ideas about what, you know, what should have happened, or what what needed to happen, better, what needed to happen on Earth for life as we know it to begin. Now, it doesn't mean it happened this way. It just means that as far as we know, with the evidence that we've got, you know, as far as with all the research that we've done and all the ways that we've looked at it, we've decided as a scientific community here that these four really major things that I'm talking about in biogenesis, those are the things that if everything happened the right way, we think we have a pathway. We say, okay, there's a logical pathway between A to B, and B to C, in this case 1 to 2, and 3, and then to 4. See, we're going to talk first about organic molecule formations. Now how many of y'all remember what an organic molecule is from Bio 1? What's the definition of an organic molecule? What makes a molecule organic versus non-organic? That's right. And what's so special about carbon? I mean, why is carbon the foundational element? I'm sure your bio teacher told you that for all the living things on Earth. Why carbon? There's 100 and something elements. And that means what, Brittany? That's right. That's 4 valence electrons. That means carbon can do what, or carbon wants to do what? That's right. It can bond with other elements, and it can bond with other elements. And how does the octet rule fit into all of this? How does that number, the octet rule, fit into how many bonds carbon can make? Okay, but right. What makes it stable? The octet rule makes it stable. What exactly happens when an atom fulfills the octet rule? Okay, so the valence shell is full. How many electrons does that take? It takes 8. Carbon has 4, so it needs 4 more. No, it needs 4. Carbon has 4 in its outer shell, right? And then it needs 4 more. So therefore, carbon can make 4 bonds. So what we mean about carbon when we say, okay, let's translate the chemistry. The fact that carbon needs 4 bonds to fill its outer valence shell. Now, I don't even remember from Bio 1, maybe you don't recall, but the reason why atoms get together and share electrons or ions form or any of that sort of stuff is all about stability. So filling that outer shell, it creates a very stable atom. It may not, it doesn't mean the charge, they come out of the box with 6 protons and 6 electrons in the case of carbon, but it's that outer shell that's not full. So from a living perspective point of view, the fact that carbon is looking for 4 other electrons, or can make 4 bonds, you can do lots of things with carbon. Oh, I'm doing that with up the nose. I'm sorry. You can do lots of things with carbon. They're like Tinker Toys. Any of y'all ever play with Tinker Toys? I don't know if anybody does anymore. Tinker Toys, those little wooden toys that had there was a round one with a bunch of holes drilled in the side and then the top, and then there was these sticks of different lengths and all that, and you could put them together and you could make everything. Look it up online, okay? Tinker Toys, you could make anything out of Tinker Toys. You could make a giraffe, you could make a boat, you could make here, I'm going to send you a link. I found the best way to kind of show you things on the web without without having to switch back and forth is to just send you all a link. Because if I switch back and forth, then what happens is I lose where I am in my place in my slides. It messes everything up. So I'm just going to send you. Click on that link. These are Tinker Toys. This is what I mean. This is the analogy I'm trying to make. The round wooden things, you could make so many things out of Tinker Toys. And the beauty of the toy was simply the way those wooden things were drilled so that they had all these places to put things. So you weren't limited to square corners or whatever. You could do all kinds of things with it. Carbon is like that little round peg there, okay? Where you carbon is like the round wooden thing. You can attach all kinds of things to it. Carbon can be attached in small molecules, large molecules, really big molecules. You can hang things off of carbon, all kinds of other. Remember functional groups? You guys remember functional groups? Name a functional group for me, somebody. Yeah, good. All right. So this one here, what's this one? That one right there. What's that functional group? Or did you already say it? Nobody said it yet. Yeah, they create alcohols. That's right. It's a hydroxyl. There you go. So so you have these chains of carbon, short, long, and remember, you can also put double bonds between the carbons, and what that does is it causes the carbon atoms to bend in a certain way. So you could actually use chemistry to shape things. You see what I'm saying? So shape things, shape complex molecules, really complex molecules, shape really complex molecules that can do metabolism. And when you got metabolism, guess what you got? because rocks don't metabolize, do they? Rocks don't metabolize. So when you get to those complex chemical reactions that you learned about in enzymes, when you talked about enzymes, enzymes are proteins, right? And proteins are organic molecules. So it's all driven by the fact that you can take an element, carbon, and because of its need for 4 electrons, you can make lots of different really complex molecules. Hydrogen ion pumps, cytochrome complexes, ATP synthase, all those things you learned about back in Bio 1 you can. Okay, so the fact that you can link those carbon molecules together is very important for this diversity of life. Now, once we have these organic molecules, then we're going to start wanting to even make them more complex, linking them together. So we're going to go from little ones to big ones. We're going to go and create macromolecules. Now, do y'all remember what's the name of that process, that chemical process by which 2 monomers can be linked together? 2 monomers to make a polymer. What's the name of that process? Yeah, now, in dehydration synthesis, or in any endergonic reaction, there is something that you're going to need. You can't just have reactants. You can't just have A and B. If you're going to synthesize something, it's an endergonic reaction. What am I saying? What is needed to make that reaction occur other than A plus B? That's right, energy. Okay, so what we're going to do when we take these organic molecules after we get their formation, we're going to link them together, and we're going to need energy to do that. And I'll show you where that comes from as we get into more. I'm getting my cup over. As we get into this a little bit deeper. All right, now after this, what happens third is what we're going to do is increase the efficiency of these really fancy molecules that are doing kind of a metabolism thing, right? We're not at life yet, so we're not going to call it metabolism, but they are just about at that point where they're being able to do that. But what we're going to do is we're going to package these molecules. We're going to concentrate these molecules, these fancy macromolecules that are doing all this, and we're going to put them in little vesicles. So the chances that the molecules that do these fancy things increases because they're in a smaller volume. All right, go back for a second to Bio 1. An enzyme and a substrate. Yeah, they need to fit together. I know the lock and key part. That's not what I'm referring to. But an enzyme is on one side of a cell and a substrate is on the other side of the cell. How do those 2 particles, how do those 2 things get together so that they can bind in the lock and key arrangement? Does the enzyme say, hey, substrate, I mean, they're molecules, right? What determines whether they get together? Okay, those are, once they get close enough, that means that lets them bond together. But I'm saying, okay, you see my hands are way on the other side of the cells in the cytoplasm. What gets them close enough to even try to bond together? You're overthinking it. No, it is passive. It is passive. If I drop red food coloring into a beaker of water, what does it do? It diffuses. It spreads out, right? And now, when is red food coloring? Is that a little animal that can swim? Why do the molecules of red food coloring diffuse? What makes them move? No, not osmosis. It's diffusion. You have that diffusion. Osmosis is a type of diffusion, but that's not what the red molecules are doing. It's a natural tendency from Mother Nature. Yeah, they're moving down a concentration gradient. There you go. It's a natural tendency from Mother Nature want to spread these things out. But what causes the molecules to drift apart, the red molecules? Just bumping into one another. molecules bump into one randomly. So how does that substrate and that enzyme get together? They're bumping around in the cytoplasm, bang, 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 and they just happen to bump into one another. Random, right? And just like we talked about with drift, what is the determining factor of drift? What is the determining factor in those two particles getting together? You know, the enzyme and the substrate. Chance. So how can you increase the chance of those things happening? Well, with the case of the enzyme and the substrate, you could heat it up, right? Remember, those molecules that are bouncing around. Well, there you go. Those molecules are bouncing around when they heat up. There you go. And then if there's more of them, Tiffany, and if we think not only just more of them, or when we think of them as more of them, we mean we package them, right? Fewer of them in a smaller volume means greater chance they're going to carry out these really cool reactions that are metabolic, like that we got going on, right? See how y'all see what I mean? So you see how we're kind of, I mean, obviously there's a lot that's going on in between each of these steps. But you see how we're kind of building the argument here for the support of the idea that this is perhaps one of the ways that happened. And then, okay, when you get to the last one, it seems to make sense. If you spent, if this effort that was gone through to make these really fancy molecules, make the fact that you're going to use carbon, oh, look at this, we can do lots of things with it. Okay, then we can make bigger molecules. And then we package that together, we can actually increase the efficiency. It's kind of like, can you imagine what would have happened if the first person who ever invented ice would have died before they wrote down the recipe? Imagine how different the world would be. First person who invented ice died before they wrote down the recipe. Come on, I know it's 2 o'clock in the afternoon, but you must be a little more awake than that, right? It's a joke. Tough audience. So no, I was just saying that, can you imagine going through all of this? Can you imagine going through all of this idea where these molecules, these fancy molecules are coming together? Now, think about one of the molecules you learned about in Bio 1 that really is important, how it's put together. You remember when we talked about the primary level of sequence? What were we talking about? Which kind of organic molecule were we talking about? We were talking about the primary level of sequence. There was a primary, secondary, tertiary, and then the quaternary. Right. And what are the monomers that make up proteins? What are they called? There's 20 of them. Nope, that's DNA, amino acids. Right. And what's important about the order of the amino acids in a protein, or why is the order of the amino acids in a protein so doggone important? What does that determine? That's right, how it folds, how it shapes. Identification, sure, exactly. See, enzyme and substrate got to fit together. There you go. Enzyme and substrate got to fit together. So there's an active site. Remember it's lock and key, right? That's a shape. That shape only comes about if you have the right amino acids in the right order. So let's say you put the right amino acids in the right order and you get these cool molecules that are doing this metabolic kind of thing, and then they fall apart because everything falls apart. It's called entropy, right? And you forget how to do it. So the last thing that's going to happen with living organisms is there's going to be this way of writing it down. There's going to be these molecules that can actually record how to do this, this metabolism that we're experimenting with. And these molecules are going to be able to make copies of themselves. Now, what kind of molecules am I talking about in number 4? What kind of molecules arose with life on Earth, that we're referencing in step 4? These ones that contain the instructions. Somebody No, no, polypeptides are the proteins. Yeah, that's right. DNA, the DNA, remember? The DNA is your genetic code. The DNA tells your body how to look, structural proteins. Remember all the functional proteins? There were enzymes, transport proteins, hormones that were proteins. There were immune system proteins, antibodies are proteins. Holy cow, proteins. And it was all about shape. The protein, I tell my students in my Bio 1, write it right across the top of your notes. The most important thing about a protein is its shape. And you only get the shape right with the right amino acids. And you better make, it's really beneficial if you keep a copy of how to do that, if you keep the recipe. And you make it self-replicating so that you make lots of these specially shaped molecules. Okay? So let's go back, and then we'll do the progression of life. After that, we'll talk about, once we get to what, you know, everybody kind of would consider life, then we'll talk about how that happened on Earth. I don't know if we'll get that today, but we will get to it next time that we talk. Remember, there's no exam next week. It's the week after. No, let me go back a second. Let's go with number one first. Number one, organic molecule formation. All right, if we look at the early Earth, this is what the early Earth kind of looked like. It wasn't a very nice place. There was a lot of volcanic activity. So there was a lot of stuff in the air. There was water. We're not sure exactly where that came from. Some folks maybe think that comets brought our water crashing. The early Earth, remember, or the early solar system was a big pinball gallery, like I said, man, thing, including comets running into each other. So maybe a lot of our water came from that. Yeah. But there's a McDonald's. So it's not so bad. So all of this stuff this is belching out of these volcanoes. And I'm not just talking about a couple of Vesuvius here and there, Krakatoa there, Mount St. Helens. I'm talking about most of the planet are these volcanoes. And not only volcanoes, but there's areas on the planet like out in Siberia. I'll show you a map a little later on. I don't know if we'll get to it today. Where like half of Siberia, you know, that big open area in Russia, half of Siberia was a lava lake. So it wasn't a volcano, it's just this lake of molten rock and boiling lava and all these gases like hydrogen sulfide and ammonia, carbon monoxide, carbon dioxide, all nasty things were coming off of this lake and coming out of these volcanoes. And so the early Earth, what was, yeah, that's right, Harrison's right, it's terrifying. We would not have survived. Living things weren't around. They might have, there's some ideas that maybe a couple of times life tried and then went extinct. That's really sort of interesting, but it was about 1 billion years after the Earth formed, so about 3.5 billion years before life actually began. So it took 1 billion years for the Earth to settle down and become a hospitable place, at least for us. See, the early Earth's atmosphere was not like ours now. See what I wrote up here? I said the early Earth's atmosphere was reducing. Now go back and remember again in Bio 1 or maybe in your chemistry class, you learned about something in chemistry known as reduction and oxidation. Sometimes you may have seen it referred to as redox reactions. Somebody let me know what that means, if they remember. What does it mean when we talk about a reduction reaction? Or an oxidation reaction? What's going on in those? Which one is which? That's right. Yep, reduction is adding electrons. Okay. Remember, it's the weird kind of word, like you're adding something, but it's called reduction. But what's going on, of course, is the electrons are negative, so it's being reduced in charge. So it makes sense there. Okay, so in a reducing atmosphere, what's going to happen is we're going to favor reduction. What kind of atmosphere do we live in now? We don't live in a reducing atmosphere. What kind do we live in on Earth? Well, unless we don't live on Earth. It's the name of our, what's the name? Yeah, it's an oxidizing. An oxidizing atmosphere, oxidation tears things apart, right? That's that whole thing about antioxidants, green tea, pomegranate, blackberries, blueberries are all high in antioxidants, which is supposed to slow down entropy, right? Entropy always wins. But here in the early Earth, or in the early Earth's atmosphere, it wasn't that way at all. Molecules weren't encouraged to fall apart the way they are now. Molecules were actually encouraged to be reducing, to actually come together. Like what? In the ozone? Yeah, yeah, that's actually part of what the UV here, like that we were going to talk about, because that's one of the things that changed the Earth too, right? So we got this reducing atmosphere, and reducing atmospheres are conducive for making bigger molecules, for making polymers. So there's this idea of making more complex molecules. Now, if before we actually get to this idea of the UV and then Bradley's question with the ozone, I want to talk for just a second about this idea of energy. Why are we saying that we need energy? So we're reducing things. We have an atmosphere or an environment that's conducive for building bigger things. Why is energy necessary for that? What's that? What's that about thermodynamics that means energy is going to have to come from somewhere? You remember when you're building up bigger molecules from smaller molecules, that's right, energy, that's right, it's neither created nor destroyed. But so when you're building up bigger molecules from smaller molecules, you've got to put in energy. It's like the analogy of you got to put in energy to roll the boulder uphill. So if you're going to be reducing, if you're going to make bigger molecules, if you're going to have an environment that's conducive for polymers, it's not enough just to have A plus B. You got to have the energy to put A and B together. Where would that come from? Well, number one, the early Earth did not have ozone. Yeah, it's transferred from other organisms. That's right. It's transferred from A to B. So you can't create it, you can't destroy it, but you always need it because you're building up molecules. Being that the fact that the early Earth didn't have much or didn't have any oxygen, there was no ozone. Ozone is O3. It's a special type of oxygen. It's actually 3 oxygen atoms. It's that has a electrical. If you've ever been around if you've ever been around sparking wires, electricity, that acrid kind of smell is ozone. If you've ever been down in SeaWorld near where it used to be, I used to walk by it all the time going to work, you could smell it because they used ozone as ozone kills everything. They used it as a purifier. They would bubble ozone gas through the water before it was pumped back in for the animals, because the ozone would bubble through. And like I said, even viruses can't deal with ozone. Good thing about ozone is it has sort of this dual life, because on Earth, it's way, way, way up, like, I don't know, stratosphere or beyond. It's way up in the upper atmosphere, so it doesn't cause us any problem health-wise. But what it does do is block out ultraviolet rays. See, ultraviolet rays are bad in they're bad in small concentrations. That's how you get skin cancer. It causes mutations to your DNA. Ultraviolet rays are very powerful. They're like Mother Nature shooting little BBs at your DNA, bing, 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 and trying to knock off an adenine or mess up a thymine or mess up a piece of your DNA there. That's what kind of UV is. Now you get UV. That's, like I said, that's how you burn at a beach. You get sunburn, even under clouds. UV goes right through clouds. So in the early Earth, there was no ozone, so there was a tremendous amount of UV. Okay, well, that obviously wasn't good for living things, but we're well before living things back then. But that was a lot of energy that could drive those reactions, those endergonic reactions to make polymers. There's also a lot of lightning. Oh, you got to check this out. Here's another link. When volcano, not just from the clouds and the water, the rain and all that on Earth, but when volcanoes erupt, you can get lightning. Lightning is caused by friction, and in rain, it's raindrops moving past each other and wind masses and ripping electrons off of each other. But it can also really happen in a volcanic cloud, because there's all these little particles knocking each other around. That creates static electricity. So there can be lots of really dramatic lightning coming off of a volcanic cloud. You guys see that picture? Click that link. Does it? No. No, that's real. Dustin. Isn't that neat? There's plenty of other pictures out there, though, too. But, yeah, you can get volcanic lightning. And lightning's got a boatload of energy in it. What, like 100 million volts or something like that? There's also these places we didn't know about till the early 70s. This is how so cool about science. I mean, I remember as a kid them finding these things, these geothermal vents. Think of geothermal vents as being like underwater volcanoes. They're basically, again, the same idea. They're down at the bottom of the ocean, and the rocks have cracks and fissures. And below, you know, I'm sure you all know the Earth is a crust that floats on top of this semi-liquid mantle of molten rock, right? Well, the water seeps down between the cracks in the rocks, and it gets heated by this mantle area, and it gets heated up to boiling. But the water didn't boil down there at the bottom of the ocean because of the pressure. The pressure's so high. Hey, that's how water boils off of your stove. You heat it up until the water molecules, they're moving so fast that the pressure is greater than the pressure from the atmosphere, right there at where the bowl is that you're heating up, and the water bubbles into the atmosphere. So if you have a lot of pressure, water can get really, really hot and not boil. So down at these geothermal vents, there's three kinds. They call them the black smokers because they look like the stuff is pouring off of them. White smokers, and I think clear smokers, and it's temperature. White smokers can be like 86 degrees Fahrenheit. Black smokers 350, 400 degrees Fahrenheit. That's right. That's why they say, oh, they used to, when I was a kid, on boxes of like spaghetti and stuff, it would say, when you're boiling the spaghetti like X amount of minutes, it would say add 2 more minutes for altitudes above 4,000 feet or something like that. Because what would happen is the water would boil before it was 212 degrees. You know, Fahrenheit, yeah. Now, these geothermal vents with all that heat, that's a lot of energy, right? And remember, the water's not boiling, so the energy not being transferred, because that's what happens when things change state. So you get a lot, especially in them black smokers, and all that heat makes this, oh my gosh, a soup of all these organic molecules. Oh man, it's like geothermal vents are some of the neatest places on Earth. Look them up. You got to look them up on YouTube. I might even have some videos that I put in Blackboard. If not, find some. Down there are critters, crabs, and different species that only occur around this one geothermal vent. That's it. And they're like 30 feet away. Sometimes they're kind of spread out, you know, across the bottom of the ocean. 30 feet away or 50 feet away, there'll be another one. And there'll be another group with species that are only found there. So like these are unique species on Earth, and like right there, that's all of them. Like if you could count them. Well, there's 8 of them. That's it on the entire planet. But all that energy down there from the heated rock and stuff would boil up a lot of material, and it could, you know, like I said, it could have been enough energy to be conducive for making these polymers. Hot sand, clay, and rock. If you got a volcanic world, you got a lot of hot sand, clay, and rock. And I'll talk in just a moment about why that's significant when it comes to making these bigger molecules. Who respirates differently? The dogs are snoring in the background if you hear them. Yeah, that's right. Yeah, Bradley, I'm sorry. Yeah, chemosynthesis. They don't use photosynthesis. There's no light down there, obviously. And instead of water, they use hydrogen sulfide, which there's a lot of. Remember what water does for plants in photosynthesis? It's a source of electrons and protons, right? Remember, it splits it up? Well, you can do the same thing with hydrogen sulfide, and what you give off is protons and sulfur. So there are critters down there that do that as well. Oh yeah, it's a neat place. Take a look at some of those videos. It's just absolutely incredible. Okay, so let's take a look at Pasteur's work. Louis Pasteur, the fellow who's also known for creating the way to pasteurize your milk to keep us safe, was a scientist, 19th century? I think it was 19th century. Early 1800s, something like that. Anyway, at that time, one of the hypotheses or ideas out there about the way life began was this one of spontaneous generation. Now, spontaneous generation basically says that non-living gives rise to living. So that, if a cow falls over the side of the road, and in a few days, the cow's rotting away, and all of a sudden you see these maggots, then you think the maggots came from the rotting flesh. That non-living flesh, non-living, gave rise to living somehow, okay? And it's easy to see why they would think that, right? Because if you walking down the road, dead cow, you might see exactly that, maggots after a few days. You got to remember, they didn't have quite the instruments to see the unseen world back then. And humans are always that way. I mean, it makes perfect sense. If we don't see it, we have a hard time understanding that it's there. I can explain to you all day long the structure of an atom, but it would sure be nice to see them, It'd be nice to go, oh, there's the nucleus, and there. It doesn't work that way. Okay, so all of those observations about spontaneous generation weren't insane by any stretch of the imagination, but Pasteur was out to set the story straight. So he was gonna either support or refute spontaneous generation. Remember, never prove or disprove a scientific theory. It always keeps our mind open. So here's what he does up here at the beginning. He takes a flask of broth, which is a nutrient broth. Things can grow in it. It spoils. And he boils it, so presumably everything's killed, and lets it sit, and then, of course, it does spoil. All right. So the spontaneous generators out there are saying, see, Louis, because he was French. The non-living broth, because you boiled it, nothing could live in that, gave rise to these living microorganisms. Now, they didn't see the microorganisms, but they saw a bunch of them. So they saw mold or crud. Stuff that sometimes I find in my refrigerator, right? Like, oh my goodness, throw that away. All right, so here's how Pasteur did it. Beautiful piece of scientific work here. Just scientific method all the way, bang, bang, bang. So here's what he does first. He takes a broth and he sterilizes it once again and he seals it. He says, okay, take a look at this. Oui. No organisms appear at all. So if you spontaneous generators are right, then, this sterile broth should give rise to spoilage, evidence of microorganisms, evidence of things that we can't see. And they're like, okay, I gotcha. Except there is one decent criticism of this. Can you guys think of what maybe the criticism could have been here? What could someone have said back then? Hey, here's what you missed about your experiment that might have biased your results. Yeah, life needs air, doesn't it? So right away you could say, well, wait a minute there, Louis. The thing you're missing is the non-living can't give rise to the living until there's air. You got to have air. Oh, okay. So what he does is he builds the next contraption that's got a swan neck. It works just like the trap in your sink. If you dropped something down the drain of your sink, then it could or would or should, at least initially, falls into the trap. Heavier things would. Microorganisms would be heavier than air. Not a lot, but they are. So that was Pasteur's thought process there. So what he does is he takes the sterile broth, puts it in the swan neck, so no problem with air. Everybody can accept back then that the air is moving through, and guess what? Still remains sterile. The microorganisms or whatever was coming from the outside to give the impression that the sterile broth gave rise to life can't get in. You see, that's very strong refutation. That's a very strong refutation of spontaneous generation, folks. Obviously, everything that's necessary for life is now being exchanged with that sterile broth, and yet we still don't have living things. Now, Pasteur does the next thing. He does the slam dunk. He says, now I will demonstrate to you that what caused the living things to appear in the broth came from the outside, because I'm going to break the top of this thing off. And now it's exactly like basically it was with the panel number 1. And guess what? It spoils. So he's like, fantastic. That's, you know, that is direct evidence to suggest that what happened is not a situation of non-living sterile broth giving rise to living things. Isn't that neat? Good stuff, Louis. So Louis Pasteur demonstrated pretty convincingly that you can't make life from non-life, right? Everybody agrees with me on that. Give me a thumbs up. Give me an emoticon. Interact. Say something. Type something in there. You all agree with me that this definitely suggests that you can't make living from non-living. Yes. Very good. Thank you. Well, you're wrong, because obviously that's what happened. When we talk about it biologically and chemically, that's exactly what happened. Now, it didn't happen this way. Remember, you can't throw away a theory sometimes because the mechanism is wrong. The idea might be right, but perhaps the wrong mechanism. Lamarck had the wrong mechanism. Right idea. Darwin got the right mechanism. Wegener, the guy who came up with this idea of the Earth being crusts or plates that float and bump into one another, he was right about that, but he had the wrong mechanism again. So the spontaneous generator, folks, so this idea that life can come from non-life had to have occurred if we look at the way, the evidence of life on Earth. Where did we come from? And if someone say, well, it came from another, where did it come from there? So it's the mechanism again, folks. Always remember that in science, you got to keep your mind open. That's why Pasteur, he refuted spontaneous generation. He refuted the mechanism, not the concept. Okay, so we're going to talk now about ways we think it may have happened. So I'm going to go back to over those 4 steps again that I outlined at the beginning, you see. First, we're going to do, I sound like the old actor. First thing we're going to do is we're going to make organic molecules. We're going to take carbon and we're going to make complex molecules out of them, right? We're going to make, hopefully, we're going to make amino acids. Because remember, carbs and lipids, while they're organic molecules, their function mostly is energy storage, long-term or short-term. It's the proteins that contain information, right? That sequence, just like the nucleic acids, it's the same thing. That sequence of amino acids is a code. So there's information there. So those are going to be much more than just a water molecule. Those are going to be much more complex. So let's start with this thing called the Miller-Urey experiment from 1953. This is a famous experiment that was done. It's been recreated many times. It's even been done in high schools and with positive results. So it's not something that was done once and then never recreated. That's always been the problem with nuclear fusion, getting that to be repeatable. But the Miller-Urey experiment, named after these two fellas, assumed what the early Earth was like and attempted to demonstrate whether you could make complex organic molecules out of it. So first we'll start up here in the upper right with what they had as an atmosphere, big globe there, right? So there's gases in there, and the gases that they put in there were things like, well, there's water vapor, nitrogen, ammonia. I'll list some of them there. Carbon dioxide. Oh, there's also hydrogen sulfide, H2S. These were gases that were assumed to be in the early Earth's atmosphere, and this has been from astronomical studies where we look at gases on other planetary bodies. This is also from, this is really cool, when glaciers form, the ice that forms is older the deeper you are in the glacier. So many times when the water freezes, it freezes bubbles in the ice, and those bubbles are bubbles of gas the way the atmosphere was whenever that ice was formed. And they can date that ice, and they can say, oh, this ice formed, several 100,000 years ago, and they know what the atmosphere was like. It also happens in rocks. So when rocks cool off, they can cool these little, they can form these little bubbles of microscopic bubbles, in some cases of gases, that are reminiscent or that are what it was then. So that's where Miller and Urey got these, you know, concepts, or at least the support for putting those gases in there. Because if not, then you're like, well, heck, you don't know what the early Earth was like. Okay, so anyway, you start that with the atmosphere, and you add some energy, because you're going to build complex molecules. You're going to go from simple to more complex, and you got to have, that's an endergonic reaction, so you can't escape energy. Thermodynamics, the laws of thermodynamics. In this case, what they used was electrodes to simulate lightning. So again, it could have been the UV or anything like that, but in this case, what they did is they, you know, acted like what, and there was plenty of lightning again, lots of volcanoes. And what would happen in the atmosphere, of course, is then, you know, the water would condense and cool, and it would rain, and it would rain down. So I'll come back to this in just a second, but let's kind of complete the cycle first, because when you rain down, eventually you end up in the ocean. The heat source, or the sun, would evaporate the water back up into the into the air, into the atmosphere. Now you do that a lot of times, many times during 1 week, and you sample down here in the bottom, in the trap, if you will, and what you get are things like organic molecules, complex organic molecules, even amino acids. You're able to create those. You can use them as code little monomers, right? Those amino acids are all a little different from each other, and however you put them together is how they fold up, and how a protein folds up is how it works. Okay, so you're able to create those types of molecules, those molecules that can remember shape, if you will. Remember, I'm using that word really, you know, stretching it. In just a relatively simple setup experiment. Now, you do that many cycles over millions of years, and you can create a lot more of these organic molecules. So the Miller-Urey experiment doesn't necessarily say that this is the way it happened, but it shows a way to at least say we can make complex organic molecules in the laboratory. We can recreate it. And lately, with the discovery of those geothermal vents, there's a lot of ideas that if it didn't happen out in the atmosphere, then maybe around these geothermal vents that I mentioned earlier, maybe that happened there. And then, of course, some folks, and to me this is incomplete explanation, some folks can discuss something called panspermia. And panspermia says that the way life got to Earth is that it was seeded by an interplanetary traveling body, a comet or an asteroid, that had the precursors of life on it. Nobody's saying necessarily life, but the precursors of life. And some of the astronomers, you know, think that perhaps meteorites or meteors could have brought amino acids to Earth. Doesn't explain where those came from. All right, so that's what panspermia is. But nonetheless, how it got here, there is at least some plausible explanation as to one of the ways that perhaps these organic molecules could have formed. Now, once we got these simple amino acids, once we have the monomers, then we want to link them together. So we need the energy to make complex organic molecules, amino acids. Then we need more energy to link those amino acids together to make a protein. And then once we get a lot of those, then we can start, you know, getting into the folding and the shaping of all of those things. But once again, what do you need to make monomers into polymers? What's that one requirement you cannot get away from if you want to go little to big? If you want to make, it's dehydration and synthesis. That's the process. Yes, that's right, Melanie. But what's the requirement? What do you have to make a dehydration synthesis reaction work? Other than reactants, you got to have energy. That's right. You got to have energy. So we now jump to step number 2. Here we are in the early Earth, and we've got this Miller-Urey experiment going on, or this Miller-Urey situation going on. Maybe down around the geothermal vents, maybe both. Maybe a few planetary bodies are adding to it as well, but you're getting this soup of these relatively simple organic molecules in the ocean. So this is an actual picture of, I think in Hawaii, of lava running into the ocean. It's really kind of cool if you watch it like on video. I mean, it just sizzles, and it's just, you can imagine, I don't know, lava's got to be, well, you wouldn't want to touch it with your finger, that's for sure. It's pretty hot, molten rock. But imagine this Earth of this soup of these sort of organic molecules, or not sort of, but these organic molecules splashing up onto the beach. Now, I'm not saying the ones that hit the lava, would have done anything, because instantly you become steam. But, as the rock cools, you got this hot sand, you got hot areas that water, this organic soup, could have splashed up on. The heat from the sand, the clay, the rock, could have been enough energy. Now, heat is not the most efficient form of energy, but it is a form of energy. So what you can do is you can imagine that heat could have brought these monomers together, and this has been done in the laboratory too. They've been able to make proteins. That's what polypeptides are. They've been able to take amino acids, drip them onto hot sand, hot surfaces, a hot plate probably in the laboratory, and been able to get them to produce or link up and produce polypeptides. So the energy necessary for dehydration synthesis came from heat. Okay, so, and there would have been a lot of volcanic activity on the early Earth, and there would have been a lot, presumably, of this soup of these precursor organic molecules. So again, not something that seems so far out of the ordinary. Now, once those bigger molecules are getting together, those are the ones that start using energy to converting energy. Converting energy. Those are the ones that eventually lead to the ones that can do things, and I'll type this in, that can do this. Typing it into the chat thing. Okay, so you remember that from Bio 1? Right? That's what's happening in respiration, correct? You're phosphorylizing an ADP. You're making a more complex molecule. So you need energy to do that, right? So these polypeptides, forming together are going to need energy to be able to do that. And when you were putting ADP and phosphate together to make ATP, you were getting the energy because you were oxidizing glucose in respiration. In respiration, you were releasing the energy, which you were using to make ATP. And then ATP released the energy when it made your muscle cells work, or the rest of your body, because that's how you spend energy in your body. All right, so it's just this transfer of energy that's occurring here, but energy is necessary. Heat is not the best form, but it would work, and it has been shown to create these polymers, even these polypeptides. Now, you remember the next step? You remember the next step after we have this soup of sophisticated molecules, this open soup? What happens next? What did we say in that summary may have occurred next? It's about efficiency. So you got this open soup of these special molecules, and they need to find each other in this vast world, in this vast soup of each other. They need to find each other. What would increase their chances of finding each other? Temp wood, yeah, Dustin, that's right. Temp wood, that would help. So let's assume, we're at a temperature, around the rock. It's pretty warm there. What in addition to that would increase the chances? And temperature increases the chances because since they're moving randomly, they move more quickly, better chance to bump into one another. But what would also increase the chances of them bumping into one another? Think about if two people were running around very quickly in a gym versus two people in an elevator running around very quickly. Which one's got a better chance of bumping into one another? How come? Smaller, right? So once you have this soup of these organic molecules, what's good? They can have or monkeying or starting with this metabolism functions. What's going to increase your chances of bringing all those complex molecules together to do this little dance that they're doing? Package them, right? Put them into a package. There you go. Put them into something we call a protocell. A protocell, nice fancy way to define it, but you know what this is. Aggregates of abiotically produced molecules. Back in Bio 1, you learned about a molecule that has a very special nature to it. It's both hydrophilic and hydrophobic. It was in the lipid chapter. It was in, I'm sorry, the lipid section, not the lipid chapter. What was the name of that molecule? What was the name of that molecule that has that unique nature of being attracted and repelled by water? Just like your laundry detergent is too, by the way. It wasn't a triglyceride. It was a triglyceride with one of the fatty acid tails taken off, and then there was a phosphate put on there. There, you're close. So it was a phosphate group put onto a glyceride. You remember, a triglyceride is a glycerol and 3, that's the tri, fatty acids. You take off one of the fatty acids and you put on a phosphate, and you have a, and my fingers in the right letters. There you go. Thank you. Faster than I am. You have a phospholipid. These little molecules that you saw all over the place in Bio 1. Now, what happens when you throw a bunch of phospholipids into an aqueous, a water, the earth environment? What happens when you throw a bunch of those molecules into a beaker of water? Okay, so the heads line up with the heads and vice versa. Draw on the whiteboard. Tail, okay, there you go. So in order for the heads to line up with the heads line up with the heads, but which way do the heads face, Melanie? The tails stay away from the water. The heads face out, right? So if you're facing out and you have to, you're kind of surrounded by water, what's going to happen with those phospholipids is they're going to form a bubble, right? And actually a bilayer, remember? They form a bilayer. And they form a bubble. They form a package. They form what's called, you can make them in a laboratory even today. This is a picture of one right here, 20 microns, that's the scale, of a liposome. A liposome is nothing that's alive, but it is a bilayer membrane. And it's selectively permeable, just like your cell membrane is. Now, it doesn't have active transport, but it can do diffusion, osmosis, and those kinds of things. It's not a living thing, but these are abiotically produced molecules, things that we can make abiotically. Phospholipid is not a living thing. It's a molecule. It spontaneously, by itself, because of its chemical nature, attraction and repulsion of water, will form a package. Now, if you package up these special molecules inside of these bubbles, then you decrease the volume. It's the two dudes in the elevator again. They got a much better chance of these special molecules running into one another. Now, here's what else they've done. This is what the picture on the right is showing you. In laboratories where they've created these protocells, what they've done is they've added a few simple enzymes, okay? They've added, for example, things in the red. They've added phosphorylase and they've added amylase. These are relatively simple enzymes, okay? They're not the most complex of enzymes. Amylase is in your mouth, actually, because the proper name of starch is amylose. And amylase is the enzyme in your mouth that starts digesting starch as you eat it. Now, into this mix added was something called glucose phosphate. Now, glucose phosphate in this experiment basically acts like ATP in our cells, the energy currency. So this is the source of energy. So what they've done is they've taken these liposomes, they add some enzymes, they add phosphorylase and amylase, they add a source of energy in glucose phosphate, and what they can get is a production, first of all, of starch. And then amylase can come along and break that starch down. Now, turn it into maltose. What kind of a sugar, what kind of a saccharide is maltose? How many monomers made up maltose? You all recall that. Would it help if I told you that it was a disaccharide? Okay, how many monomers are there in starch? How many saccharides are there in starch? Or what's another name for it? Many, right? Many. Can be huge. Starch molecules can be massive. So we're going from a big molecule, starch, using the enzyme amylase to break that down into maltose. What kind of a reaction is that when large is broken down into smaller? Endergonic or exergonic? And we're going from a bigger molecule to a smaller molecule. It's an exergon. Energy is released. Now, isn't this, in essence, in the very simplest form, isn't this what you're doing in digestion? Aren't you breaking down starch? Aren't you breaking down glucose in respiration? Aren't you breaking down larger molecules into smaller? You're releasing energy, and you're using that energy to do this, to drive an, it's like one gear turning another, remember? Whoops, wrong arrow. That's what you're using the energy for. You're using the energy to drive that reaction. And then the ATP, that energy is being spent in your muscle cells. And does it get recycled? Well, it doesn't get recycled, but it's there because it's the heat that's released, right? Your energy, your muscle cells contract, and the amount of energy that isn't turned to work, because it's not 100% certain, is the heat that's released off of those kinds of reactions, those muscle cells contracting. So if you take these liposomes, these abiotically produced molecules, and just spike them a little bit with just a couple of simple things that may have been around, or a couple of simple fancy molecules, these polypeptides that we're forming, and some sort of a source of energy here, some chemical source of energy, you can make these things undergo very basic metabolic reactions. Very basic metabolic reactions. Now, in a huge soup, how many of these bubbles would actually, have the fortuitousness to encompass the right amount of enzymes and to be in the right condition? Very, very few of them. Very few of them. So out of a billion of these soup bubbles, or billions and billions and billions of them, a mere handful would probably have the right combination of these chemicals. But now what they've also showed is that if you add something that was very common, or is common in volcanic activity, this montemorillonite clay. You're thinking, what the heck did he just say? It's up here. This is from the weathering of volcanic ash. If you add that to a solution of these liposomes, they can undergo, not really reproduction, but they can start budding off considerably faster than if you leave them without. See, in the red is just the precursor molecules for making these liposomes. The blue is you put in the precursor molecules for making them, and you add a little bit of this clay. And that stimulates the formation. The y-axis is turbidity. Basically, the more little bubbles or vesicles, the cloudier the water is going to look, right? That's what turbidity means. Turbidity is cloudiness. So if you just sprinkle a little bit of that clay in there, you really bump up the formation of these little bubbles. It's the same way they get the rain to form in clouds with airplanes. Airplanes will fly through a cloud, and they seed. They seed for the rain. They drop out these little particles, and the water droplets, the minute little microscopic water particles, start sticking to this molecule, or this little particle. It gets bigger and bigger and bigger until it finally gets heavy enough it falls as rain. Same thing you're doing here. You're causing these abiotically produced molecules, these phospholipids, to want to clump up into these vesicles. And you can see in the blue, you speed up that process considerably when you have like this stuff from volcanic ash, which I don't want to say again, because I've said it 4 times today already. It's a tongue twister. All right, now look at the picture down below. That if you throw in a little bit of RNA, and we'll talk in just a minute why that's not DNA, but if you throw in a little bit of RNA, look what you get. Again, an actual picture. Those vesicles start taking up that RNA. All right, now we got these special molecules. Now we have RNA. What happened to DNA? Well, we think the world initially started off as an RNA type world. We think the world's first self-replicating molecule. We think the world's first information-containing molecule was RNA. You remember DNA and RNA, that's what they do. They contain information. DNA, A's and T's and G's and C's, and then RNA, you know, through transcription. That's just information. That's how do you make a protein? You start with this amino acid, then this one, right? That's information. It's the code. So RNA is also a molecule that has information on it. So, but the difference between RNA and DNA is double-stranded. And the reason why we think RNA was first was we think the first molecule that came about just stored information, a sequence of bases. We know this because there are molecules that are still around today known as, like, for example, there's the hammerhead ribozyme. That is a sequence of RNA that acts like an enzyme. It actually stimulates chemical reactions. DNA came along later on, we believe, as a more permanent copy of the code. You remember, the DNA doesn't get recycled. RNA does. You unzip the DNA, you make a piece of messenger RNA, you send it on out to the ribosome. The ribosome translates that then to a sequence of amino acids, and then the messenger RNA falls apart. The code is maintained in the DNA. So the DNA is like the archived version of your code. RNA is single-stranded. There's better chances of it being monkeyed with, of it falling apart, of getting messed up somehow. So DNA with the other strand kind of helps seal that code in. It's really kind of simple if you think about it, the difference between the two molecules and the need for them. But back to this idea of ribozymes. These ribozymes, like I said, they exist today. These aren't things just from the past. It can actually catalyze reactions. So they're just sequences of RNA. They're adenines, guanines, not thymines, uracils, cytosines, and they can, you know, begin to catalyze reactions that we would consider metabolic reactions. And then because this was a sequence of letters, then that could be inherited. And if this molecule was self-replicating, it could be passed down. And if it sounds like I'm adding more and more characteristics of life, this organization and metabolism, reproduction, and all sort of stuff, it is because we're getting to that point where we're getting off of the non-life into the life. When does that exactly happen? I'm not really sure. I don't think it does have an exact spot. All right, so, you know, from the beginning, what we did is we have organic molecule formations. Then we take those organic molecules and we stick them together. We make them bigger because we want to make them more complex. Then we package them to increase their efficiency of contacting one another, these very complex, very shapely molecules. And then we remember the code by which to do that. And we pass it on. We replicate it. To me, understanding the sequence of the way life occurred on Earth is like coming into a movie, like a 3-hour movie, in the last couple of minutes, sitting down and trying to figure out the plot. Right? We, as humans, come on the planet very late in life, very late in the life on Earth. All right, I want to cover these next two slides rather quickly because I'm running out of time. You guys have seen this before in Bio 1. This is how and we talked about it before. This is how we date the fossil record. It's all about the radioisotopes. The amount of parent versus daughter isotope concentration tells you how many half-lives it's been dead. So if you don't remember what that was, please go back and take a look at your Bio 1 information. So if in the case of dating the rocks, the half-life is an order of billions of years. That's how we date the stuff that's really old. See, carbon-14 is only good for about 75,000 years, back to about 75,000 years. Dinosaurs, you couldn't date the dinosaurs with carbon-14. When you date the rocks with dinosaurs, when a dinosaur completely fossilizes, its bones become rock. That piece of petrified wood is not wood anymore. It's rock. It just replaced, mineralized the organic material, to almost perfectly like the wood itself. But dinosaurs, you're dating the rocks around them, and you can date the rocks in the same way you date, things with living things. This carbon-14. It's all about these radioisotopes. Those radioactive isotopes decaying, and they decay at a certain rate, that half-life. And if you know how much is in your sample, then you know how many half-lives it's been. See, like, for example, living things, we no longer accumulate carbon-14 after we die. Most of the carbon we take in, we eat, is regular old carbon, 12? Some of it's carbon-14. When you die, that starts decomposing because it's radioactive. You can figure out how long someone's been dead by looking how much is left. So someone, if you examine some organic material on them, you know, some mummy they find, and 50% of the carbon-14 has decomposed into this daughter isotope, you know that it's been dead about 5,700 years. Because the half-life of carbon-14 is 5,730 years. Okay, review that if you don't remember. If you got any questions, then by all means talk to me. So that's how we're getting these dates that you see on this scale. You know, 4.6 billion years, the origin of the Earth. The oldest rocks about 3.8 billion years. So, you know, the Earth was around, you know, before we can actually date it back to the oldest rocks, because we can look at other evidence in the solar system. First, it was the prokaryotes, and this, which we'll talk about, changed everything, man. Let me tell you. When oxygen began to rise in the atmosphere, then the eukaryotic cells, they started to really proliferate. And then these really sort of, these, we don't want to call them simple, let's call them ancestral critters, were moving onto the scene. The jellyfish, the sponges, or I remember like my zoology professor used to always say in college about sponges. He was from Kentucky, so his accent, he'd say, they do so well with so little. Right? That sponges, before we say they're primitive animals, let's accept the fact that as a sponge, they do a really good job, and you might actually be fascinated by sponges when we get into them. But here's Darwin's problem. Here's the Cambrian explosion. And look what happens in the Cambrian explosion. The sudden increase of all these animal forms, for reasons we'll talk about when we move on. We get a lot of marine algae, and then we start venturing out onto the land. First by the fungi, then the plants. Actually, by the fungi and the plants together, actually. Then the animals. The animals need something to eat. And then we'll talk about what vascular plants are. But plants are getting more complex, and along come the animals. They're getting more complex now. We're getting fishes. We're getting four-legged critters, insects. They were very early. Insects were one of the most prominent organisms out there, you know, pretty much have remained that way. The Carboniferous period is interesting. It's named that because when we look at the rock layer between 299 and 359 million years ago, we see this heavy amount of peat, of organic material. The world was very wet during this time. There was tons of forests, all kinds of these vascular plants, ferns, some of the first seed plants. There were some reptiles crawling around. The amphibians were going crazy because this was perfect for them. It was wet, moist, lots of wet areas. Remember, amphibians got to go back to the water to lay their eggs. So this was like the amphibians' heyday. Devonian is usually considered the age of the fishes. Carboniferous is the plants and the amphibians. We move on and things start to dry off a little bit. We'll look at that later on, but about 250 million years ago, the land masses on Earth were forming, and that was forming a lot of dry areas. So the reptiles, with their amniotic egg, began rising to prominence. Insects were moving out on land. There was a big extinction at the end of the Permian too, and that continent was formed, which we'll talk about when we get to that part too. So then we move, of course, in the Mesozoic, the Cretaceous, the Jurassic, the Triassic. You always remember the Mesozoic as the era of the dinosaurs because, of course, the movie Jurassic Park. That's when the dinosaurs were abundant. That was their heyday, that 50 or so million years in the middle of the Mesozoic. At the beginning of the Mesozoic, excuse me. At the beginning of the Mesozoic, plants moving out on the land was stimulating this evolutionary adaptation of plants being more careful about their seeds, so protecting them at things like cones. Dinosaurs were evolving and radiating, but mammals were too. Mammals didn't come after the dinosaurs. They were evolving during the dinosaur time. They had to suffer through that middle period when the dinosaurs were the dominant ones. These cone-bearing plants continue to dominate, and then we move, looking at plants and moving beyond cones to fruits. Fruits are very beneficial for plants because it's an incentive to spread the seed. Dispersion is very important. 65 million years ago, the dinosaurs, here comes the asteroid. It's all over for them. And then the mammals. Mammals and the leftover dinosaurs or the leftover reptiles, the birds, the pollinating insects. And the pollinating insects correspond with the flowering plants. Who pollinates flowering plants now? It's the insects for the most part. Some of it are wind pollinated, but the insects. So there's this idea that organisms evolve in a really major symbiotic way. They kind of evolved together. You know, as I was saying, animals couldn't have moved out onto land until the plants did first, so there was something to eat. So these flowering plants, these angiosperms, their dominance in the Eocene was because of this pollinating insects. Mammals continue to evolve, primates, more mammals, and then we start showing up, first of all, as bipedal human ancestors, not humans, and not humans till around somewhere, 2.6, 2.5 million years ago, along with the woolly mammoths. So here we come in at the last minute of this movie and try to fill in all those details. It's a daunting task. It's really what keeps science so interesting to me. We know we're never going to answer all these questions. And to me, that's the most fascinating part about this. All right, so next week what we'll do is we'll continue on with this idea of how life on Earth, once it began, and I've summarized what we see here, we'll go back and start looking at it in a little more detail. What were the events that were changing the Earth, the climatic and the organic, the biotic events that were changing the Earth, okay? So let me stop the recording.